Okay, good morning, friends and dear student. We are in TYBSC Zoology class, paper one semester, paper two semester one, and title of our paper is Mammalian Histology. In this, today we will discuss 3.5 point from the syllabus, that is a gland associated with digestive system. In this, we'll see that salivary gland like parotid, submandibular and sublingual glands. But before directly going to detailed histology of salivary gland, we must know basic histology of gland. So with some few slides, I will try to discuss with you that basic histological idea of what is that gland is. So any gland is an organ that consists of specialized secretory cells. Now here in many histological section we have seen that there are many different cells, mostly squamous epithelial, cuboidal epithelial, columnar epithelial. So but here when we say it is a gland, it contains a epithelial layer which has a secretory kind of cells. This secreted material is usually a liquid. They all the time they when we say it's a gland, it produces a liquid secretions that could be a mucus, serous or enzymes, but it is in a liquid form. They all are epithelial in origin. All these glands secretory cells are epithelial cells. They present in single, either it is a single secretory epithelial cell works as a gland, or it is a group of cells working as a gland. Here they are either unicellular or multicellular. So further, these glands are mainly classified in two types according to their way of secretion, or that secretion, the way of secretions, how it get mixed with the blood. So it is exocrine glands and endocrine glands. So in our body, in mammalian system, you find that glands either they are exocrine or endocrine. Endocrine glands, you are well familiar with the many endocrine gland. Thyroid is endocrine gland, and here salivary gland is the exocrine gland. Now, when we say that in unicellular glands, a single cell distributed among non-secretory cells. The example is that goblet cells. We have seen that histology of trachea, in that epithelial layer of a trachea and windpipe in respiratory system. We say that there is a mucus secretion goes on. That mucus secretion is because of this goblet cell. So now it is a unicellular gland, a gland made up of a single cell, and that single cell is responsible for secretion of a lot of mucus, here in this case called as a goblet cells. And goblet glands or goblet cells, if it is a goblet gland, then it is a unicellular gland. Now multicellular glands, here many secretory epithelial cells pour their secretion in a cavity, and there is a duct of a epithelial cells which also are in a secretory functioning. They form a duct, and that secretion is then collectively collected and poured outside. So the many secretory cells work as a gland here. So it is a multicellular gland. They form by the invagination of epithelial layer in a deep, deeper tissues to form a diverticulum. Now here many such epithelial cells group together and they form a diverticulum where the secretion is stored and poured outside. That is proximal part that forms the duct. Here the initial part of that diverticulum, they form the duct system or tube system, and the distal end portion part forms the main element, secretory element. So here the secretory unit, the example is a acinus. Acinus is a group of gland which produce secretion in their lumen, and that get transferred through the ducts. Now, the one more point here they required for exuding their secretion is they require a lot of muscles in their cellular portion. So These epithelial cells are myoepithelial cells. Then they have intercalated ducts. So these are all intercalated ducts. In every acini, they are connected with some duct, and duct also formed by the secretory epithelial cells. Then there are striated ducts and excretory ducts. So with the chain of this duct, that secretion get poured outside. Here, if you try to compare this exocrine and endocrine glands, so the major concern is they pour their secretions on the surface of epithelium directly or through the ducts. Okay, we have seen an example of goblets, so they directly pour their secretion on the surface, or seen that example. Now we are going to see that example of a salivary gland where saliva is poured through the duct system into the mouth. Here in case of endocrine gland, they are called ductless because their secretion is directly poured into the blood. The blood capillary are very closely attached with these glands, so the secretion is directly entered into the blood system. So these are the ductless glands or endocrine glands, and here these are all exocrine glands where the secretion is poured on surface through the duct. Now here you can see all these are the secretory cells, and the secretion is poured into these capillary portions. Now here the secretion is poured into the lumen, the central portions, and then through this duct system it is poured outside. Now if we try to classify these exocrine glands, they are based on the shape and branching pattern of a duct, or they are based on the mode of release of their product as merocrine, holocrine, apocrine. Then based on the nature of their secretions, what kind of secretion they produce, acidic, alkaline secretions, and on the basis of shape and branching pattern, the shape on alveoli, tubular, column shape, on that basis you can classify this exocrine gland. And here today's our topic is a study of a histology of salivary glands. In that there is a parotid, submandibular and sublingual. These all come under the exocrine glands. So we are revising this topic. Okay, so based on the shape of secretory units, tubular and alveolar and branching pattern of duct is there. So here you can see a simple and compound glands. Simple gland where only a single lining of a epithelial cell is there, and a single duct pour their secretion outside. So simple glands and simple secretory unit on based on the their arrangement. Here in compound secretory units you will find that a single tube gives lot of branching and branching and branching, and they form a network, and the secretion is poured into the smaller tubes, then collected in larger tubes, and then collectively poured on the surface. So simple compound gland, they are tubular, simple cuboidal tubular, simple branch tubular, and simple branch alveolar. Now the variation is either they are coiled tubules, a simple straight tubule, or coiled tubule, that is spring-like structure, or branch tubular. Few branches are there, and this is a alveoli with the branch. So simple branch alveolar. And in case of compound glands, you will find compound tubular tubes. Lot of tubes are there with the branches, and compound alveolar. At the end of the that compound gland there is a alveoli, and compound tubulo-alveolar. Lot of tubule as well as alveoli are there. That is a compound tubulo-alveolar gland. Now simple gland again classified as a alveolar. This is a acini forming gland. A single acini, a single lumen and small duct form through because of this acini, and there is a secretion poured on surface here. So this is acini is a ball-like structure, capsule, like grape-like structure, internally lined with the centrally pointed epithelial secretory epithelial cells which pour their secretion here. Now in the tubular, they goes deep, forming a long tube, and that tube is surrounded with all secretory epithelial cells. So there is no as such cavity is formed or lumen is formed in between the tubular gland. This is a tubular alveolar. So initial part forms a tubule and the latter distal part forms the capsule or hollow space, bulge or alveoli. So this is a tubular alveolar glands. These are all simple glands because there is no branching here. Now same alveolar, tubular and tubulo-alveolar glands are there. Only the exception is there is a lot of branching happens, and that is the characteristics of this compound gland. So here you will find these are the alveolar glands, but they are united with many different alveoli forming the branch network system. Here they are tubular, but they forms the branch network system. Here again they are alveolo-tubular, but forms the branching networks. So these are the compound glands of alveolar, compound glands, tubular and tubulo-alveolar compound glands. Here you can see the examples of different these simple and compound glands. So crypt of lubricant in a wall of a digestive system, intestine, that is simple tubular glands. Then sweat gland present in a skin. They are simple coiled tubular. You can see the tube is coiled here. Then simple branch, a single one or two branches are formed. So that is found in a stomach fondic glands. So this is simple branch tubular gland. Here simple branch acinar. Many small acini are there, but not very much branch. All the acini unite together and pour with their secretion through the single duct. So that is a simple branch acinar. Meibomian glands. Example is the Meibomian glands. Here now these two are the compound glands. You can see here Brunner's glands, submandibular glands and mammary glands. Among that we are interested in this submandibular glands. Here to see here there is a tubular glands. The secretion is in the form of a tube and they are connected through long network of ducts and poured outside. In case of this, they are alveolo-tubular glands. So there is a formation of a small alveoli, then the tubule and the network of branches. And here, mammary gland is a purely acinar compound gland. There is less number of tubular glands are there. Majority are alveolar. So lot of alveoli, grape-like structure, bunch of a grape-like structure you see here with all that branches, and here the branches are like a tubes and they pour their secretion outside. Now these glands are also classified on the basis of their mode of secretions. So how they pour their secretion outside. On that basis these glands are classified as merocrine glands. These merocrine glands secrete or release their secretion through exocytosis on the surface. Apocrine glands, where that part of a gland get detached. The part of a cell stored all that secretion and get detached from the original parent cells, that is apocrine. Holocrine, here the entire cell is shed out with the secretory product. Example is in a sebaceous gland. So here in diagram you can see this is a simple alveolar gland showing the merocrine secretions. These dot, all these small granules are the secretions and that represent the secretions, and that secretory granules are released on the surface through exocytosis. That vacuoles come to this cell wall surface, cell membrane, and that membrane then produce exocytosis and release the secretion in a lumen, and then it goes outside. So secretion is released by exocytosis of secretory granules. For example, in a pancreas, here exocrine secretion of pancreas, then parotid gland, which we will study soon, and the protein component of milk from the mammary glands is released through these merocrine type of secretions. In apocrine, apical portion of the cell here the anterior portion of a cell is directed towards the lumen. That's entire portion of a cell which secretory product is pinched off, get disconnected from the original cell and released into the secretion. So it's a lipid component of milk from the mammary glands. So in mammary gland, you will see both merocrine as well as apocrine gland if you study the histology of mammary glands. Then holocrine, here the entire cell. So see here the cell goes on accumulating the secretory product, and as that secretory product matures and that cell get full with the secretory solutions, that entire cell get detached from the basement membrane and released into the lumen. That cells then goes outside and burst. So entire cell is shed along with the secretory product, that is called as a sebaceous. You will see that example in a sebaceous gland. The mode of secretions. So merocrine secretion by exocytosis, like this. You can see here how that vesicle comes to the cell surface and release their content outside. That is a merocrine secretion by exocytosis. This apocrine secretion, here the secretion by loss of the cytoplasm and that burst and the release secretions. Holocrine, the secretion through loss of entire cell. So classification based on the nature of secretions. We are still in the we are still discussing about the classification of cells. Glands, classification of glands. Already we have seen classification based on the shape and then the type of secretions. Now here nature of secretions: serous glands, mucus gland, and mixed gland. So what they are releasing outside, the chemical nature of what they are releasing outside. So here the serous glands secretions is a thin, watery, and rich in enzymes and protein in nature. So most enzymes are secreted in this kind of a gland. Then cytoplasm is a granular that stain darkly from pink to dark purple with hematoxylin eosin stain, and they are rich in rough endoplasmic reticulum. So here all these dark color structure represent the serous glands because their granules are darkly stained with the hematoxylin eosin stain, and their nuclei are rounded and basally placed. So their nucleus you will never see in directing in the lumen side, but their nucleus is always arranged at the periphery of that acini. And so this kind of glands which present such structures, they are called as serous gland. Then mucus gland, here the secretion contain mucopolysaccharides which collect in the apical part of a cell. So the chemical nature of a secretion is a mucopolysaccharides, and they are collected in the apical part of the cell. So all that secretion is stored in this apical portion here. The cytoplasm stain very lightly. In previous serous glands, you will find that stain that hematoxin eosin stain it very dark. If it is a mucus gland, it is stained with very light color. So all these are the mucus glands, and you see the nucleus, but not you will not see the much more stained color in a central portion of this centrally directed epithelial cells. So light color stain because with the hematoxin eosin stain. That is the characteristics of a mucus gland, and their nucleus are basally placed and flattened. Mucoid pushes the nuclei. So, see, nuclei are more flattened and basally placed. In earlier serous gland, we have seen they are very round but basally placed, but round insecta. Mixed gland, here you will find some are serous producing, some are mucus producing. So there is no such distinction you will see here. Now in this color photograph or here somewhere, if you carefully observe, some are dark color structures and some are light color structure. Okay, so these are the mixed color and contain both serous and mucus secretory units. Okay, So these are the serous dermaculins, and these are the mucous dermalidin. Sometimes serous cells forms concentric caps on the mucous acini called as the serous dermalins. These are the serous producing cells over the this serous mucus producing cells, all centrally located. They are mucus producing cells, and the surrounding are the serous producing cells, and they are overlapping over the mucus producing cells. So these cells are called as the serous demulins. Now here in color photograph we'll try to locate that serous demulins. So this dark portion here on the side of these all acini, here everywhere. Okay, in every acini you will find this some dark portion. So we will call this as a mixed gland because it shows mucus gland as well as serous gland together.